Hi, I'm teaching AS level biology under the AQA specification, and today I'm teaching transport across cell membranes, which is part of the cells topic. So in 1872, the fluid mosaic model was proposed to describe the structure of cell membranes, with fluid, meaning phospholipids form a bilayer in which the phospholipid molecules are constantly moving, and mosaic, meaning there are proteins of different sizes and shapes embedded into the phospholipid bilayer, so it looks slightly like a mosaic. Here you can see the structure of the membrane. And we have the phospholipid bilayer, which is made up of phospholipids with hydrophilic heads and hydrophobic tails, cholesterol, which is a protein that may be intrinsic or extrinsic, glycoproteins, which are proteins attached to a carbohydrate, and glycolipids, which are lipids attached to a carbohydrate. The lipid molecule is made up of a hydrophilic head and two hydrophobic tails. This causes phospholipids to arrange themselves in a bilayer so that the hydrophilic heads are facing out towards water and the hydrophobic tails are facing in, away from water. This arrangement creates a hydrophobic center in the bilayer so that water-soluble substances cannot pass through. However, lipid-soluble substances can dissolve in the bilayer and pass directly through cell membranes. Cholesterol molecules provide stability to cell membranes. So cholesterol molecules consist of a hydrophilic and hydrophobic region. The hydrophobic regions bind to phospholipid fatty acid tails, causing them to pack more closely together. Proteins in cell membranes can be grouped into two categories, intrinsic and extrinsic proteins. So intrinsic or integral proteins are embedded through both sides of the phospholipid bilayer, and these include channel proteins and carrier proteins, which transport large molecules and ions across the membrane. And we have extrinsic or peripheral proteins, which are present only on one side of the phospholipid bilayer and provide support to the membrane or may be involved in cell signaling. We have glycolipids and glycoproteins. So glycolipids consist of lipids attached to carbohydrates, and glycoproteins consist of intrinsic proteins attached to carbohydrates. So glycolipids and glycoproteins are involved in cell adhesion, which is the attachment of cells to one another, cell recognition, which allows cells to recognize one another, and cell signaling, which is the communication between cells. Now on to the roles of cell membranes. So there are two main types of cell membranes: cell surface membranes, which surround cells that act as a barrier between the cell and its environment, and they control which substances enter and leave the cell. And we have membranes around organelles, and these surround organelles, such as mitochondria, to act as a barrier between the organelle and the cytoplasm, dividing the cell into different compartments. Both types of membrane are partially permeable, meaning they let some molecules pass through, but not others. Now we're on to diffusion. So diffusion is the net movement of particles from an area of high concentration to an area of lower concentration. Particles diffuse down a concentration gradient and eventually reach equilibrium, in which all particles are evenly distributed but still moving. Diffusion does not require energy, so it's described as a passive process. There are two key types of diffusion: simple diffusion and facilitated diffusion. Some molecules can diffuse directly across cell membranes in a process known as simple diffusion. These can be molecules such as oxygen and carbon dioxide, because they are small, which means they can pass through spaces between phospholipids, and nonpolar, which means they can dissolve in the hydrophobic core of the cell membrane. Larger or polar molecules can cross cell membranes in a process known as facilitated diffusion, which involves two types of proteins: carrier proteins and channel proteins, which are both highly specific, allowing only one or two types of molecules to pass through. Carrier proteins mainly transport large molecules across the cell membrane, and so a large molecule attaches to a carrier protein, which causes the carrier protein to change shape. Then the carrier protein releases a molecule on the opposite side of the membrane. Channel proteins mainly transport ions across the cell membrane, and these proteins form pores in the cell membrane which ions can travel through. So the factors affecting the rate of diffusion are temperature, which at higher temperatures particles have more kinetic energy and diffuse faster; concentration gradient, the steeper the concentration gradient, the faster the rate of diffusion; the thickness of the membrane, so the particles travel shorter distances through thin exchange surfaces and therefore diffuse faster; surface area, so larger surface area means more particles can cross the membrane at once, which makes diffusion faster; and the number of carrier or channel proteins, so the more of these proteins, the faster the rate of facilitated diffusion. Now, osmosis is the diffusion of water molecules across a partially permeable membrane from an area of high water potential to an area of lower water potential. Water molecules are small and can diffuse directly through the cell membrane, whereas larger solute molecules cannot. Water molecules diffuse down a water potential gradient until the water potential is in equilibrium. You can see in the image there, water potential is depicted by that little trident symbol and is measured in kilopascals. So solutions are made up of solvent dissolved in water. Water potential is the pressure exerted by the water molecules onto the membrane or container surrounding a solution, and like I said, is measured in kilopascals or kPa. High water potential means the solution has a high water concentration, and low water potential means the solution has a low water concentration. Pure water has a water potential of 0 kPa. This value decreases as the water potential lowers. Now focusing on animal cells. So when animal cells are placed in solutions, water may move into or out of the cells depending on the water potential of the solution. So in hypertonic solutions, the solution has higher water potential than the cell, which means water molecules move into the cell and the cell swells and bursts. In isotonic solutions, the solution has the same water potential as the cell, which means there's no net movement of water into or out of the cell, and the cell stays the same size. In hypertonic solutions, the solution has a lower water potential than the cell, water molecules move out of the cell, and the cell shrivels up and shrinks. Now, in plant cells, unlike animal cells, they do not burst due to the presence of a cell wall. So in hypertonic solutions, when the solution has a higher water potential than the cell, the water molecules move into the cell swells and becomes turgid. So, in isotonic solutions, the solution has the same water potential as the cell, which means there's no net movement and the cell stays the same size. In hypertonic solutions, the solution has a lower water potential than the cell, which means water molecules move out of the cell and the cell shrinks and becomes plasmolysed, which is a word you can only use for plant cells. There are 4 factors affecting the rate of osmosis. So there's temperature. So at higher temperatures, water molecules have more kinetic energy and diffuse faster. Water potential gradient, which means the steeper the gradient, the faster the rate of osmosis. The thickness of the membrane, so water molecules travel shorter distances through thin exchange surface areas, therefore diffuse faster. And surface area, so larger surface areas mean more water molecules can cross the membrane at once, making osmosis faster. Now, onto active transport. So, active transport is the movement of particles from an area of lower concentration to an area of higher concentration. This process requires energy from respirations in the form of ATP, or adenosine triphosphate. Energy is needed as particles move against or up the concentration gradient, and therefore active transport is described as an active process. Like facilitated diffusion, active transport involves the use of carrier proteins to transport molecules or ions across membranes. So in this process, the molecular ion binds to the carrier protein, ATP binds to the carrier protein, and the hydrolysis of ATP to ADP and phosphate causes the carrier protein to change shape. This releases the molecular ion on the opposite side of the membrane. Then the phosphate is released from the carrier protein, which causes the carrier protein to return to its original shape and is ready to be used again. There are 4 factors that affect the rate of active transport, so we have temperature, and at higher temperatures, particles have more kinetic energy and travel faster. Respiration also increases with temperature. However, very high temperatures denature carrier proteins and decrease the rate of active transport. The thickness of membrane means that particles travel shorter distances through thin membranes and travel faster. The number of carrier proteins, so the more of these proteins, the faster the rate of active transport. And the rate of respiration, so the more respiration, the more ATP available for active transport. So now I'm going to be talking about the co-transport of glucose in the ileum. So some carrier proteins combine to 2 molecules at once, and these molecules are known as co-transporters. They use the concentration gradient of one molecule to move the other molecule against its own concentration gradient. And this occurs during the absorption of glucose. So during digestion, carbohydrates are broken down into glucose. The glucose is absorbed from the lumen of the small intestine into the bloodstream. However, diffusion can only result in equal glucose concentrations, so not all the glucose will be absorbed. To overcome this, glucose is absorbed from the intestines into the blood using co-transport. So the diagram shows the ileum, which is the final part of the small intestine. As food passes through the ileum, the glucose passes from the lumen, which is the space inside, into the epithelial cells which line the ileum, and finally into the blood. The co-transportation of glucose involves three different proteins: sodium-potassium pumps, which actively transport sodium and potassium ions, sodium-glucose co-transporter proteins, which use facilitated diffusion to transport sodium ions in glucose molecules, and glucose protein channels, which use facilitated diffusion to transport glucose molecules. So glucose is absorbed into the blood through the following process. Na+ is actively transported out of epithelial cells into the blood by the sodium-potassium pump. This pump transports 3 sodium+ ions out for every 2 potassium+ ions in. This creates a concentration gradient, as there is now a higher concentration of sodium+ in the lumen than in the epithelial cells. Then, Na+ diffuses from a high concentration in the lumen to a low concentration in the epithelial cells. It's transported via sodium-glucose co-transporter proteins, which also carry glucose molecules. This causes the concentration of glucose inside the epithelial cells to increase. There is now a higher glucose concentration in the epithelial cells and in the blood, so glucose diffuses out of epithelial cells into the blood via facilitated diffusion. Glucose molecules are moved against their concentration gradient due to the sodium+ concentration gradient, rather than by using ATP, so they're moved by co-transport rather than active transport. The same process is used to absorb amino acids in the small intestine, where amino acids take the place of glucose. However, ATP is used to maintain the concentration gradient of sodium ions between the lumen and the epithelial cells of the ileum. Thank you! Bye!